Good morning. I am Aymer Vasquez from Universidad del Valle and International Center for Tropical Agriculture. My presentation is Molecular Taxonomy of Pulp weevils, Rhynchophorus and Dynamis, and their geographical variation in morphology and genetics. Rhynchophorus and Dynamis are Dryophthorinae weevils. These share a cleaved antennae and a division of the genitalia. Both genus have a similar shape and color. However, the males present hairs in the beak of the Rhynchophorus and the femur of the Dynamis. In addition, Dynamis has a posteriorly extended pronotum and Rhynchophorus a bigger scutellum. Both genus are pulp border of the stems and inflorescence respectively, belonging to the complex of the pulp stem border. Rhynchophorus is the most dangerous and Dynamis is a new pest in Colombia. Rhynchophorus and Dynamis attack pulp with economic impact, such as coconut, dates, oil pulp, fish pulp, hair of the pulp, and ornamental pulp. Rhynchophorus has 9 species from all continents except Antarctic. There are geographical patterns of the species in the different continents and morphological variation, for example in Rhynchophorus palmarum and Rhynchophorus ferrugineus. For this reason, the molecular taxonomy is a new approach in the group. The more recent analysis showed a good delimitation of the species with the COI gene. Despite this, there are few sequences for R. palmarum and R. phoenicis and some problems in the delimitation of R. ferrugineus and R. bilineatus. The barcoding analysis demonstrates geographical pattern in the north and south population of R. vulneratus in the same way R. ferrugineus has geographical pattern, for example, in China, is north-south and east-west direction. Finally, in R. palmarum, there is a differentiation in some locations in eastern Brazil. With morphometry, it has only been described for R. palmarum. In this case, the north and south population also is delimited. Dynamis is a neotropical genus with 3 species follow a new taxonomy according to morphology. Also, there are more species described. Dipirupacus is in Central America, Diboras is in Central and South America, Dinitedulus in Amazons. There are only 3 sequences for Diboras in the gene bank, including one for the COI genes. Our goal are assessing the support of the Rhynchophorus species with COI genes, describing new COI sequences for Diborasi, identify the geographical pattern of the Rhynchophorus clades using COI genes, and describing the morphological differentiation of Diborasi and R. palmarum of the southwestern population in Colombia. We use samples from 30 locations, all coming from the Pacific, Andes, and Amazon in Colombia. For the molecular analysis, we use the larvae and adults. For morphology, only the adults. The specimens are in the entomological collection of the International Center for Tropical Agriculture, Colombian Sugarcane Research Center, Cenicaña, and Universidad del Valle. The DNA was extracted in the 10 specimens with the CTEB methods. We did the PCR amplification for producing COI fragments using the universal primer. Moreover, we cloned this fragment with plasmid DNA and Escherichia coli. Finally, we sequenced these new cloned fragments. Later, we used all COI sequences of Rhynchophorus and Dynamis from GenBank database. R. ferrugineus was the largest represented, followed by the other species of the Rhynchophorus and Dynamis boraci. We aligned 1,849 sequences, including 11 related species, as an external group. Next, we identify 132 haplotypes, and finally, we calculate the best evolutionary model and construct the phylogenetic tree with a maximum likelihood. We evaluate this tree with a bootstrap method of 1,000 replications. For the morphometric analysis, we measure 19 characters of the head, thorax, and abdomen, similarly to previous study of the R. vulneratus and R. palmarum. The samples were organized in 4 populations according to the natural region and altitude, corresponding to Amazon, Pacific, High Andes, and Low Andes. The high Andes has 1,000 to 1,500 meters above sea levels, while the low Andes is a valley of the Cauca River with 900 to 1,100 meters above sea level. The measurements were organized in a database. As well, we calculate the principal components. Finally, we compare the population with discriminant analysis. The 7 species studied were monophyletic with bootstrap support greater than 80%. This support is maintaining despite the inclusion of more sequences and new localities for most species. It was not possible to determine the relationship of these species except for R. vulneratum and R. bilineatus. Further, we detect the internal clades in R. vulneratus, R. ferrugineus, R. palmarum, and R. bilineatus. In R. vulneratus, the clades are like reported of the Rudman-Jones and collaborators in the north and south. In this case, we estimate a genetic distance of 0.04. In R. ferrugineu, we're reporting only 2 clades, separated by 0.21, corresponding to Egypt and the rest of the distribution. This result is different to the Rudman-Jones report because we used shorter COI sequences in order to analyze the sequences of R. palmarum from Brazil. For R. palmarum, the presence of the clades are a new report. In this case, the population of Western Colombia and Mexico are a clade with a genetic distance of 0.05 respect to the other clades of Esther, Colombia, and Brazil. It is possible that the Esther mountain range of the Andes in Colombia divides these populations. Moreover, the morphometric analysis of Rhynchophorus palmarum demonstrates that the low Andes population is different to the other. These results were evidenced in the discriminant analysis. It should be noted that Rhynchophorus palmarum in the low Andes is mainly associated with a sugar cane. In the other population are mainly associated with palms. The attack in this population are concentrated in young sugar cane with little food for the weevils. It is possible that this weevil modified its larvae adult development times with consequence on sites. In population of R. ferrugineus was documented the influence of date palm variety on the size of the weevils. Another singularity of the low Andes population is the lack of the dimorphism with respect to the other population, which have a strong differentiation. This study is the first multivariate analysis of the sexual differentiation and comparison in different geographical populations in this genus. In a different way, Diboraci did not present a geographical differentiation. However, there is evidence to suggest morphological differentiation according to the host in sympatry. In the Amazon, one female specimen in the black boss was collected in a pitchpalm crop in similar way to the other specimen in the black boss. The other 2 specimens in the red boss were collected in a nearby locality without pitchpalm attack. These specimens are possible to associate with other pine in the forest. This situation was repeated with male specimen from Pacific population, with 3 specimens from the forest and its native palm in contrast to the other from pitchpalm. These results are related to the previous report in R. palmarum in sugarcane because Dynamis use the inflorescence and the stem of the pitchpalm while only the inflorescence in the native palm. Like to R. palmarum, its most population, Diboraci present a strong dimorphism. In conclusion, we found that the 7 species belong to Rhynchophorus and Dynamis boraci study were separated with COI genes. Also, R. palmarum population from the west in Colombia and the ones in the east have differences in the COI genes, as well as R. palmarum population from valley of the Cauca River are morphologically separated from others in the southeastern Colombia, possibly related to the use of the sugar cane. Finally, the Diboraci population present a morphological differentiation in sympatry, possible mediated by host palm. We appreciate the funding from the Ministerio de Ciencias in Colombia, also to the Universidad del Valle and International Center for Tropical Agriculture, particularly to Inge Ambrecht, James Montoya, Wilmar Torres, Julia Mendivil, Tatiana Valle, and Caterina Castillo, and all farmers which collaborate in the field work.